all in vertical 2 of DDH training program. So today's speaker is Dr. Abhishekh Singharoy from Arizona State University. I request Mr. Khirod Buragohai to kindly introduce our today's speaker. Over to you, Mr. Khirod Buragohai.
Thank you, Devabro. Abhishekh Singharoy is an assistant professor in Biodesign Institute of School of Molecular Sciences at Arizona State University. His research is at the confluence of molecular biophysics, hybrid modeling, and large-scale computer simulation. He combines rigorous statistical mathematical methodologies with state-of-the-art computational and big data approaches for capturing cell-scale biological responses with atomic precision. A major focus of his group endeavors is to seek hidden chemical information from low-resolution cryo-electron microscopy datasets. He is a co-developer of Popular Molecular Dynamics Engine NAMD. The group is developing small molecule and peptidotherapeutic interventions in mitochondrial disorders and aging, and T-cell responses to viral infection, including SARS-Cov-2. Singharoy is a recipient of the Beckman Fellowship, Sciolog Fellowship, and the NSF Career Award. Sir, it's over to you for presentation, sir.
Okay, thank you. Thank you for the kind introductions, and thank you all for coming. I'd like to thank the researcher who had nominated me for this talk, Gautam Mukherjee. So with that, then let me start. So today we're going to talk about resolving uncertainties of structure-based drug discovery with molecular dynamics. So as was already said, so let me first tell who we are. So we are a 2 years old group. We are a very, very new research team. We started around 2018 in the state of Arizona, USA. And so we have a fair number of graduate students and postdocs and some funding organizations that I'd like to acknowledge. And with that, then let me begin what we do. So in a nutshell, what we do is we try to make what you call a computational microscope. So, of course, we all know microscopes. They like to show us things in very gory details and very high resolution. We tend to like to see high resolved biological objects on a computer. So in order to do that, what we do is there are a lot of experimental methods that are high resolution. Experiments like NMR, X-ray crystallography. Now cryo-EM is getting there. There is FRET, which is using fluorescence, electron fluorescence. So we end up taking experimental data from these modalities and try to combine them with molecular modeling. And once we do that, a combination of experimental data with molecular modeling gives us very detailed insights on things like on biological functions, things like membrane permeation, molecular motor action, large-scale movements inside the cells, in fact, going up to cell divisions. One of the most recent examples that we are very, very happy about is we ended up making the first all-atom model of an entire cell organelle. That's what you see here. It's like a molecular model of an entire cell organelle composed of proteins embedded in membranes. And this is a particular one that absorbs sunlight. It's a photosynthetic organelle. So yeah. That's what this sort of modeling allows us to do. If you take data from different modalities and try to combine them using physics, using molecular simulations, you end up getting these sort of large hybrid models. So this is this area of what we call hybrid modeling. That's where we are. So that's the general focus of the group. More focusing on today's presentation. Let's now get into drug discovery. So as we all know, as is the theme of this meeting, so there are different stages of drug discovery. There is target selection, there's lead discovery, lead optimization, preclinical development, and then finally clinical trials. So of these, particularly during the lead discovery phase, I would say there are two particular steps, which is testing for potential binders and validation of their mode of action. In these two steps, computational chemistry plays a major role. And often it's very, very well known that computational chemistry starts with, it's good to have well-established structures, X-ray crystallographic structures, so to speak. If you have all major drug discovery pipelines would like to begin with very high resolution structures of the drug bound to the proteins. If that doesn't happen, which is often the case, then we look into other modeling endeavors like docking and things like that. But to start out, we'd like to really have a good model. And X-ray crystallography has been instrumental in offering these models over the years. Very recently, of course, cryo-electron microscopy has been picking up a lot of speed in this molecular biology, structural biology realm, in studying the protein conformations, and there are a lot of takers, a lot of customers of electron microscopy for drug discovery. A lot of people believe that this technology can actually be extended to drug discovery, and that's where we have started. My group has started to work towards. Is it possible? Electron microscopy in drug discovery. So let's see what are the differences between crystallography and EM, and try to kind of come up with the pros and cons. What are the advantages and disadvantages of each method before we move into the drug discovery, kind of methodology. So yeah. So then, just to start out, so crystallography, the general workflow in crystallography is you have this X-ray diffraction data that you can get from experiments. So if you go to the protein data bank, any file that you see comes up with the coordinates, that's your PDB file, but at the same time you get files like the CIF files, the CIF files, which actually contains within them the diffraction data in forms of amplitude. So if you have this amplitude of diffraction, you need an initial model, because you need to generate what you call the phase. You have to resolve what you call the phase problem of crystallography. Crystallography gives you only a diffraction of the problem, not the phase. You need a trial model, an initial model. And using this initial model and the amplitude from the diffraction data, you can actually end up constructing an electron density. Then, of course, within this electron density, you fit an atomic model, you fit your initial model into this, and then you get a refined model. Once you have a refined model, there's a convergence, there's a tolerance test. You compute something called an R value. The R value is a measure of how closely is the structure representing the electron density. If the R value is lesser than a particular value, then you say that, okay, fine, now less than tolerance, then you say that that's your final structure. However, if it's not, then you reiterate. So the procedure looks like this. This electron density is something that's evolving. It's not a constant value. So the phase is getting refined, and as the phase gets refined, the density it evolves from a poor density to a very, very well resolved density. And see, finally you end up getting, let's say, a well resolved structure. This is a low resolution density, so it's not as resolved, but you end up refining the densities. That's the point. You don't have a fixed density. You end up refining your density, and of course, the effect is both local as well as global. Much unlike this, cryo-electron microscopy, the experimental procedure is actually you generate a sample, then you put it on this grids, that's what you call it, freeze your sample actually. Then you take it inside electron microscope. India at this point of time has developed its electron microscopic facilities, and they are doing pretty well. So once you have your electron microscopic, in an electron microscope, what happens is you end up taking two-dimensional images of your sample that originally looks like this. Then you have to do something called 2D classification, where you take all your 2D images and you kind of you go over them one at a time. In a way, they're like millions of particles, often goes up to millions, like, definitely hundreds of thousands. Definitely you start out with thousands of these images, and it can go up to millions. You take all these images. These are all 2D classifications. So you can think of, you have taken photograph of a biological object from all sides, hopefully, and you have like picked up the various, orientations and the various faces of that object. Now from this 2D images, from all sides of a particular object, you can construct a 3D density. There are a lot of established tools to do it, and I'll give you an example of one that we have started developing. And then this is a nice problem to solve, going from 2D to 3D. So you have it's like you go to Qutub Minar and you take photographs from all around Qutub Minar, and then you are asked to reconstruct a 3D image of Qutub Minar. That's what it happens here. And then from the 3D maps, you end up constructing a three-dimensional model. And like the final step looks like this. You have a density envelope and you have a trial model, and the model kind of is allowed to fit into this density. So now there are some advantages and disadvantages of this technique. So the first thing is there's no phase problem. The phase problem is inherently solved in cryo-EM. Unlike this iterative refinement that I showed you in the previous slide, where the structure needs to be refined again and again, you don't have any such problem. You have the density already, and you just fit a structure to it. You can even build structures into it. No crystallization is required. No lattice artifacts are present, unlike in crystallography. Large disordered proteins can actually be dealt with this system. Unlike crystallography, where still a lot of work needs to be done to crystallize large proteins, disordered systems. Here it's not that bad. Even though this time the sample preparation time is often long. And there is, of course, after the sample has been prepared, in crystallography, it's very, very high throughput. Once you know your crystallographic conditions, you're done. Like after that, you can literally run, thousands of crystallization and then like refinement in a matter of days. Here, that's not so simple. This particular step, even after parallelization of computers, takes major amount of time, takes months, in fact. So going from here to here, going from sample to 3D map takes roughly 6 months in the best of labs around the world. So a good structure often takes, I would say 7 to 8 months to come out. Of course, nowadays, a lot of SARS-CoV-2 structures, people have been able to manage to solve them in a matter of months. But the teams who are working, actually, people with history can do that. With experience, this procedure can be made faster. But normally, I would say an average would be definitely in the order of months to years. Crystallography, you can do it in a matter of days even, once the crystallization protocol is resolved. The other thing is, of course, the biological objects will have to be big, 500 kilodalton or bigger. And the even bigger problem is limited resolution. Unlike crystallography, which actually is quite high resolution, there is a graph over which cryo-EM has evolved, and it's not nearly as high resolution. It's like it used to be quite low resolution, 6 to 7 angstroms back in the day. In 2000s, things started looking better. So we started getting into sub 5 angstroms. Then this beautiful camera started coming in, high-speed cameras around 2013. And there was a drastic decrease in the resolution of all the entries. Like say 230 electron microscopy database entries were like less than 5 angstroms. And so now this is 2018. In fact, in 2020, people have even shown chemical resolution with cryo-EM. But I would still say most of the most of the EM deposits to protein data bank are in the order of 3 to 5 angstroms. There are a lot of structures coming between 2 and 3. But yeah, so now again, within 2 and 3 angstrom structures, it's something that's high resolution for cryo-EM still remains low resolution for crystallography. So tools will have to be developed in order to take this 2 to 3 angstrom resolved protein data and start doing drug discovery with that. And that's what we will get into, drug discovery innovations. So then I will kind of from here on start giving little ideas of the methods development that has been happening in my team. And, so all of them have been published. So I will not go into gory details of every method, but give you a flavor of what is possible today. And so you can so we can start thinking in these terms. That's the idea of this talk. It's more like a catalog of different methods, and you can pick and choose between them. Okay, then let's look into the protein conformation space. What are the methods in cryo-EM for protein for determining protein conformations? So the first I would say workhorse for doing these sort of drug discovery with cryo-EM is what we call molecular dynamic simulations. So molecular dynamic simulations are, just a brief introduction, is you solve Newton's laws of motion. If you have an n-atom system, you literally compute the force on every atom by taking a derivative of the potential. This is what you call the force field. People have spent decades, at least 30 years worth of work has gone in converting very high-end quantum chemical calculations into empirical force fields, into classical force fields. And then once you have that, you take a derivative of that and you let your system relax to its low energy states. And that's what it looks like here. Like you have a protein that's folded. You can start out with a random coil structure that's like that. And then you end up solving Newton's equation, and the protein will start sliding down the energy landscape into an energy minima, or a bunch of energy minima actually, and finally get folded. So the tool that we develop, and it's quite popular, it's called NAMD or NAMD. So NAMD has recently released its version 3. It's extremely fast. It works very nicely on graphical processor units. It has both CPU and GPU capabilities. CPU historically, NAMD has been working very, very well on its CPUs. Our latest version is extremely good with GPUs. And so yeah, I invite you to have a look at this paper to introduce yourself to a lot of various methodologies available for doing molecular dynamics of proteins and protein small molecule systems. And of course, there are tutorials available. You can click on this link and you can find it. So good. So molecular dynamics is one methodology that is the core of integrated modeling. Now, when we move to cryo-EM, what we do is, as I said, you end up solving the Newton's equation, and your potential energy function that I told you, it's called the force field. Let's call it UMD, that is coming from the quantum chemical calculations, the computational chemistry calculations. So we make adjustments to the force field by bringing in the electron density map as an additional potential. So what happens is, because of this additional potential, now the structure conforms to a low energy structure, but that is limited by the boundary conditions of the electron density data itself. So the data kind of pulls the structure inside it by making this force field adjustment. Again, it's this method, electron microscopy, it's called molecular dynamics flexible fitting. It was originally introduced by Klaus Schulten and Hakim Frank, who got the Nobel Prize in 2017. It was originally created in 2008. I didn't even begin my PhD then. Okay. And then when I started working with Klaus around 2013, again, this method is also very, very popular right now in the cryo-EM community. It has its own tutorial. You can have a look. So when I started with Klaus around 2013, we brought in a new version of this method. It's called resolution exchange MDFF. So what happens is, like simulated annealing in the temperature space, you can take this density and you can make lower resolution maps, and then start fitting your structure to lower resolution densities, and then gradually refine the density and the structure simultaneously. So it's like this methodology helps really in fitting, in resolving high resolution cryo-EM data. At low resolution, traditional MDFF is very, very good, but at high resolution, which is like 2 to 3 angstrom resolution of cryo-EM, traditional MDFF doesn't work as well. And therefore, you can take large proteins and you can do this resolution exchange MDFF, where you do but you're doing a simulated annealing, but not in the space of temperature, in the space of density. This paper also, has been quite appreciated in the community, and it really helped techniques like molecular dynamics graduate from low resolution cryo-EM to high resolution cryo-EM that is of importance for drug discovery. Then, of course, nowadays, as you can see now, even the side chains are very, very, well defined from cryo-EM data, looking like the crystallographic data in a way. In fact, nowadays you can even see a hole inside this side chain for certain kinds of densities. So moving on, nowadays you can not only start fitting structures, you can fit in coils, like you can literally fold proteins inside cryo-EM density. In the previous example, I showed you large-scale structural deformations where the secondary structure was still conserved, but it was still like, the tertiary structure of the protein was changing. Nowadays, you can actually fit and fold simultaneously. So this is a tool that we're calling cryofold. It's developed with the father of protein folding, Ken Dill. And so you can do it for both soluble proteins and also for transmembrane systems. This particular part is hidden inside the membrane. I'll show you more examples later. So nowadays with cryo-EM, you can actually not only do protein refinement, but you can do protein folding. So this is the first example of doing data-guided protein folding. So this paper is out for reviews, but a bioarchive version exists. Now we actually have a number of ways in which we can start with protein structures and start fitting them or building them into cryo-EM densities of high resolution. So we know how to now generate protein structures, but how good are these structures? So often one asks the question, we have gotten structures, crystallographers have been asked the question of gotten structures, how good are these structures? One way of finding that is by looking into the Ramachandran space and see, how well behaved are the backbone and the side chain, basically the backbone atoms. And if the backbone atoms are not so well behaved, then they actually form outliers on the Ramachandran space. And subsequently, the side chains also play, do not work as well. So for this particular system, we found that there are a lot of Ramachandran outliers that are coming from the loop regions of the protein. But primarily from the secondary structured region, things are looking well behaved, as good as crystallography in a way. Also, when we do molecular dynamic simulations, the quality of the results often depend on what force field is used. The famous force field CHARM, developed by Martin Karplus, Nobel laureate of 2013, and done several things, has literally given birth to the field of molecular dynamics. There are different versions of CHARM. There's OPLS that is often found inside Schrodinger tools. So we found that these sort of refinements at the level of bond distances and bond angles so this is bond angle data, this is bond distance data, give very, very similar results. The force field dependence is not as much. However, if you look into more details, the force field dependence comes in primarily in this, in what you say Ramachandran outliers. So the dihedral angles are often the sources of problem. And in our hand, inside MDFF, we found CHARM36M to be a very, very dependable force field for doing structure determination of cryo-electron microscopic data using molecular dynamics as a methodology. This has been published in 2018 now. And so, good. So then the force field dependence is not that bad. All the force fields do reasonably well. CHARM36M is doing just a bit better. Of course, after you have done force fields here, you want to know how good your model is in addition to the geometry. So it turns out that you can consider a density to be a collective variable, and you can start constructing the free energy required to deform a density from one form to another. It's called a collective variable Kohlvar or an MDFF Kohlvar. So collective variables are often used to determine free energy because free energy needs a reaction coordinate. What reaction coordinate would you choose to determine free energy? So now in very, very recent work, and this is now available in NAMD, collective variables from densities can be used to determine free energy. It's available on a nightly build. Someone can pick up NAMD and do these calculations. And so while you do real space refinement, while you resolve these densities, you can actually compute the free energy cost of a conformational transition seen here from during doing MDFF. And here we simply show that this is a reversible trend for a particular protein called adenylate kinase. So it's a free energy methodology that is that's now that the manuscript is under review. We got good review, should be accepted soon. So you can actually determine free energy cost, and you can actually learn something about the biophysics of the proteins. How popular are the open states and the closed states? Is the open state low energy or the closed state lower energy? And accordingly, you can determine some sort of drug discovery pipeline, focusing on the open versus the closed state. So this is, of course, all data from modeling. Thereafter, now let me show you a new methodology. So again, Hakim Frank with Abbas Armas. So Abbas is in Wisconsin, University of Wisconsin. Hakim Frank is in Columbia University. So what they said is, okay, free energy as an object can be determined from molecular dynamic simulations. We have done that forever now. Can you obtain free energy directly from experimental data? If you with a little bit of understanding of cryo-EM, the first thing one realizes is, as I already told you, that there are millions of particles, millions of two-dimensional images of the object. As I told you, as I gave you the example of Qutub Minar, there are millions of images of Qutub Minar from all different sites. Of course, Qutub Minar doesn't move. Fortunately, it's stationary, but the biological object is moving. So when you take these two-dimensional images, inherently you create something like an ensemble measurement. your two-dimensional images are an ensemble measurement. When we do molecular dynamics, the reason we do molecular dynamics is we want to create a molecular ensemble from which we want to measure statistical properties. In a manner similar, your two-dimensional images can be thought of as an ensemble, not an ensemble of structures, rather an ensemble of images. And then using that ensemble, I will not get into the details. The original paper was published in 2014, where Hakim, Frank, and Abbas showed that you can actually make molecular movies directly from data. This is a ribosome, this is a very subtle motion that you see, very low resolution also back in the day. As I told you, like these are old days of cryo-EM. These were old days of cryo-EM, where molecular movies could be constructed directly from experimental data. So this methodology, very recently, we have worked with Hakim Frank and with Abbas Armast, and we took several parts of this overall methodology and inserted molecular dynamics simulations in there. And with that, what we see is this paper is currently accepted to Nature Communications, and there is a small bioarchive link that you can look into right now for unpublished. So you can actually get all atom trajectories. You can actually literally combine this manifold-based machine learning. So I didn't tell you, the methodology that they had developed was a machine learning methodology where you take these two-dimensional images, cluster them using machine learning, basically find various low-energy states inside your ensemble of 2D images, and then reconstruct 3D densities. So you can actually construct not just one density now, but you can construct a series of 50 densities, and then you can see molecular movies in all atom details. This is what you call an RYR receptor. It's an ion receptor. You can see the opening and closing of that. This is an overall picture. And consistent with that, you can actually see a very up close and personal. So there is a small ion binding pocket here in this protein. This pocket can be revealed in gory details while you're looking at the large-scale conformational transition, and I'm showing you the nanoseconds of the molecular dynamics movie here. So you can like literally have a multi-scale picture of both the larger-scale deformation and the small-scale deformations, as well as an energy. I won't get into too much details here, but these are energy landscapes. So you can literally figure out how much energy is required to bind and unbind, in this particular case, a magnesium ion to this magnesium transporter protein. So directly from cryo-EM, you can determine molecular movies and your realizations of binding and unbinding. And MD simulations are, so basically you combine that with MD simulations, you get a complete picture. So okay, enough on and not only a complete picture of the structural changes, but also in terms of energy. Energy is very critical. It's the quantity that tells you how good your estimate is, your structures are. If you see too high energies, then odds are molecular dynamics hasn't given you a good pathway. It hasn't discovered a good pathway for you. Good. So then this was about the protein space. Now let's quickly move into the small molecule space. This is work done with Chris Rowley in Canada. One thing right away when you start looking into the drugs and like drug-like molecules, by small molecules inhibitors, you want to do molecular dynamic simulations of them. The first thing that you find is that the parameters, as I told you, the classical parameters for bonds and angles are good, but for dihedral angles, that's a major problem. In fact, if you see the potential energy surfaces of a molecular mechanics force field, often you find them to be an overestimate of the correct quantum mechanical estimates. But of course, you cannot do quantum mechanics because they are extremely, of course, you can do quantum mechanics, not that you cannot. But in these biological systems, when you're running with literally thousands of conformations, it's very difficult to find the right balance between, like the right quantum chemical method. It's very expensive. And in fact, this is a problem that we can see across all sorts of molecular MMFF, OPLS, you just name it. That the problem is there. It's reported by Friesner, Rich Friesner, Professor Friesner, of course, is one of the founders of Schrödinger. So then how do you address this problem? So again, now you have this problem is addressed by something called neural network potential. So what you do is you take your system, and let's see, the movie didn't play. Let me go back. Yes, so you take your system and you break the system into small chunks. You do high-level quantum chemical calculations in each one of these small chunks, and then you fit the results of those quantum chemical calculations, instead of fitting them to positions, angles and dihedrals, which is the normal way of doing doing force field creation, you actually fit them against what you call symmetry functions. The symmetry functions looks like this sort of Gaussian functions. There's also cos-like functions, and you find products of them. There are 5 different kinds of symmetry functions that can be used. So you can reconstruct you can train rather than constructing. So that now the phrase becomes training. You can train your energy functions by using this symmetry functions, which are, as I said, Gaussian-like functions or sine cosine functions, instead of fitting them. And then you can do like literally thousands of very high-end QM calculations and fit the potential and train the potential energy landscapes by using this Gaussian functions into a neural network potential, or you call it ANI. So almost means the same thing. And so there are so many symmetry functions, like 768 actually, that can be used in order to create one potential energy surface. And then people have been doing that really, really well. And so what they have found is for drug-like molecules, this ANI functions, they manage to recover crystallographic poses without even any initial bias. So you start out with a random conformation of your drug molecule inside a protein pocket, and then you end up recovering the crystallographic docking poses for a number of different PDBs. And red is CGenFF, so this is like traditional molecular mechanics, which doesn't fit as well. Blue fits better inside these densities. I'll show this to you in the next slide. So now then, there is this technology that we are calling neural network potentials, which are very, very good for treating the ligand part of your problem. And for the protein part of your problem, as I already told you, we have information from the classical force fields, as well as we have information from the electron microscopic data. So can we combine all of that? Can we combine electron microscopy, traditional charm force fields, and ANI? And the answer is yes. So what you can do is you take any protein like that, in an apo state, hopefully. Then on that you can run molecular docking. We often run glide for that. Once you have done molecular docking, then you can do your molecular dynamics flexible fitting, because you have now your protein, you have your docked ligand, and you have some information about the density. These are low resolution densities. And then once you do MDFF, finally we have come up what we call a hybrid protocol. It's an NNP charm MDFF interface. So in this particular interface, the ligand is treated using NNPs, neural network potentials. The protein is treated using traditional charm potentials, and the entire operation happens inside MDFF. So you simultaneously fit the protein as well as the drug to the electron density, to the low resolution electron microscopic density. You can reuse the same interface to do a higher level calculation where you replace this NNP charm with QMMM MDFF. And so basically you have different layers. So first of all, you do your molecular docking, then you do your this is like an MM interface inside MDFF, where this MM is really well trained. This is like very well trained QM trained MM interface. Then you can do a QMMM MDFF. MDFF is, as I said, is molecular dynamics flexible fitting a structure and your density simultaneously to the structure, protein structure and the ligand structure simultaneous to the density. And then after that, you do model quality like correlations, energy analysis, how good the geometry is, ligand strain energy. I'll show you ligand strain energy is a very, very good indicator of how good a model of a protein ligand model have you gotten. If it's not very good, if it's very high strain structure, then you go back again and restart. And then you finally, if the strain is low enough, then you get your bound model. So not only docking, this is basically data guided docking. That's what we have. So this interface is available on VMD. So VMD is this tool that we use to kind of generate all the input files. So we can literally have there's this MDFF QMMM interface present. The same interface can be used for running ANI or NNPs, neural network potentials, inside MDFF. And these are the different levels of theory. There's a way of generating the input files. The QM part can be done on tools like Gaussian, Orca, MOPAC, depending upon the level of theory. And all of this can be done inside NAMD right now. The only thing external to NAMD is your favorite docking tool. It can be Glide. It can be a tool from BioVia. After that, everything is inside. The sampling part is inside NAMD, all inside NAMD. Okay, so here is an example. This is this drug that is like not very, very well fitted to the density. And the cross correlations, these are the local cross correlations. Blue means like all blue means really, really well fitted. This yellow means low, not so well. The scale is kind of getting, it's not there on the slide, but then you'll see that. So after certain amount of fitting, the protein is now inside the density, and by the time the simulation is over, the protein is the ligand is very, very well fitted to the density. The cross correlations are quite high, and you can track that, monitor that on BMD. So it took 900 picoseconds of MDFF to resolve this ligand inside the density. So again, as I told you, so what is the quality, the measure of the quality of a ligand inside the protein, hidden inside the density? One thing is what you call the strain energy. The strain energy is the energy of the ligand in the protein complex minus the energy of the ligand conformation in water. So you take your ligand inside the protein and release it in water, and you see it relax, and you see how much energy it releases. If the ligand is really, really strained in the protein pocket, then it will release a lot of energy. If it doesn't release as much energy, that means the ligand was quite happy inside the protein pocket. So the first thing that I wanted to show was like the NNPs, this interface where you have the proteins described by charm and the ligands described by some sort of neural network potential or some sort of low-level quantum chemistry. You see that the neural network potential does so well that it finds for you pi stacking, which is otherwise not possible in PM6. The reason for that is the neural network potential is trained on very high-level theory. Even though it's a molecular mechanics tool, it's trained on very high level of theory, so it in a way acts like a QM method. The other thing, of course, is once you are done with these sort of calculations, the strain energy calculation, of course, you can do with very high level of theory. Here we have done DFT. But the surprising thing, and we found that indeed, as the structure kind of got fitted into the density hidden inside the binding pocket of the protein, the strain has actually gone down. So it's a well-fit model. It's not only a quality of fit, but also the quality of the model itself. The model is a low strain model. It's not only fitted well in the density, but it doesn't have too much strain, unlike the initial structure. The more interesting thing that we found is, trend-wise, very low-level molecular mechanics energies gives you very comparable trend as very high-level quantum chemical calculations if you are interested in finding ligand strain energy. So once you have done all the calculation and found your ligand in the best pose, you don't need possibly, like I shouldn't say this as a general statement, but in our hands, what we have found is very, very low level, like 5 minutes calculations, 5 minutes per structure gives you ligand strain energies that are in trend qualitatively similar. Both are going down as high level quantum chemical methods. So the quality, the analysis of the quality of your bound pose can be really, really done in a matter of minutes. This doesn't take hours. This particular part of the calculation will take, I would say, half a day. After Glide has done its job, it literally takes half a day to find out good ligand poses inside proteins, docked inside proteins. And then after that, it takes minutes to figure out how good are these poses. What's the strain energy? Of course, if you want to confirm that, then it takes days for some of these calculations to go through. But there is an overall prescription that can be done. And this work is now published in JCIM. I think that was already there in the slide before. You can have a look. Okay, so now the impact of all of that. Where do we apply all of this? So again, SARS-CoV-2, as I had promised. So using this method of manifold-based machine learning, now what we have is multiple different conformational movies. We want to generate a minimum free energy landscape of the spike protein conformational transition. So there are two-dimensional images available in the protein data bank and in EMDB data bank. So what you can do is you can take those images and you can make molecular movies of them, and you can get a free energy landscape. The interesting thing about the free energy landscape is most of the traditional methods like cryosparks, like Reliant, get structures that are the lowest possible free energy. However, there can be drug binding pockets, cryptic pockets, if I may, that can be there in the higher, like still low energy. So blue is low energy, it looks like. This is like the closed state and this is the open state. But this is the closed state, this is the open state, two low energy states of the spike. But there are other high energy intermediates that we are seeing right now that have not been resolved using the traditional cryo-EM tools. computational cryo-EM tools. And we want to see if these can be used for drug discovery, just to give you a bit more example. So how to do that is, first of all, we started using this BioVia tools to reconstruct a complete SARS spike model. The models that were available in PDB were all truncated. So you take this model then, and then you do this this free energy methodology that I showed you to figure out what are the different modes of motion. These are motions projected along the X and Y dimension, and this is a motion that is projected along the Z dimension. And then you combine these two motions on to find different atomic. So you first find out movements along this dimension or this dimension, and then you find what are the combination of movements along this minimum free energy pathway. So there, of course, you need to do a multi-map fitting. You're simultaneously fitting to two different cryo-electron microscopic densities in order to resolve structures within the free energy landscape. And so that's what we are doing right now. And we have, so by using the data that was originally used for getting just 1 to 2 models, we have been able to generate 59 different models of the SARS-CoV-2 protein, of the spike protein. And not only that, we have been able to basically put the glycans, solve them, and make complete models. And now we're seeking for these cryptic binding pockets inside, which were not known before. These pockets, which were not known before from cryo-EM data, you could have still seen them in normal molecular dynamics, but there was no way of validating those cryptic pockets. But now we actually have a way of seeing high resolution, high energy, high resolution SARS-CoV-2 structures, high energy intermediates, from which the drugs can access, but normally you would not be able to see them in traditional high probability structures. So that's our story for the spike protein. Now moving on to the envelope protein, another key drug target. So SARS-CoV-2 envelope protein structure is not known. There's a model that is known for the SARS-CoV-1, but then we constructed using iTaser a homology model of SARS-CoV-2 envelope protein. There was some NMR data available for SARS-CoV-1, and then we made a SARS-CoV-2 envelope protein. We constructed an all-atom model, which is a homology model, and then we refined that against cryo-EM. I'll show that to you in the next structure. And then that's the first step. So we constructed this protein SARS-CoV-2 envelope. The primary difference between SARS-CoV-1 and SARS-CoV-2 is that there's a particular helix, that a particular paddle domain that looks like that was embedding inside the membrane in SARS-CoV-1 envelope. This thing looks like floating on the surface of SARS-CoV-2. And that is because this particular region is highly amphiphilic. It would actually be on the surface of the membrane. This is the membrane. And then with this structure, and there was also some cryo-EM data, this structure kind of gelled well with a monomer of SARS-CoV-1 that was already known. So SARS-CoV-1 has two structures. One is like a monomer, and the other one is a tetramer. So the monomer and the tetramer had two different conformations. In the monomer conformation, indeed, the paddle helix was actually parallel to the membrane. Here, this thing was kind of impinging into the membrane, but then we found a SARS-CoV-2 structure that was looking like the monomer, but the monomers were looking like the SARS-CoV-1, and the rest of they were. And the other thing was, yes, there's a lot of unstructured part in SARS-CoV-1. In SARS-CoV-2, you actually have a lot of structure in the proteins down here. The reason for that is a very unique kind of an interaction, which unfortunately this particular slide, but there is a very unique interaction down here, hydrophobic interaction actually. That is very, very cool to SARS-CoV-2, which is not there in SARS-CoV-1. Of course, then after that, we moved into this protocol. Now here on, it's all classified data, so we cannot tell you what kind of drug candidates are used. So we have done a lot of drug library screening. Then we, of course, did this glide docking, figure out places where the drugs can dock. What are the drugs? Drugs don't ask me right now. I cannot say. I'm under basically a non-disclosure agreement. This week we have so the computation, and there's of course experimental data. Cryo-EM has been done also to kind of it's low resolution, but it gives us confidence on the nature, on the quality of the model, envelope protein model that we have, and so in vivo testing starts tomorrow. It's not yet day here. So tomorrow here, starting tomorrow, in vivo testing of these drugs are going to happen. But again, it's just the first part, nothing to do with patients as of, but we'll have to do the toxicity and things like that. So this is work with the Mayo Clinic at Arizona. It's one of the very prestigious medical school here. So work very closely with them. So as you have understood by now that it's very, very important to work in collaboration with experimentalists, with even though we're modelers, we work very closely with doctors and with experimentalists so that we can come up with plausible interventions for this. And now let's move on. In the last 5 minutes of my talk, I will show you vaccine discovery. I know this is a bit off topic, but again, the technologies that has been used for drug discovery can also be translated into vaccine design. So of course, vaccines, the kind of vaccines that we are all very, very aware of are what we call B-cell vaccines, which is what generates antibody responses. We are very, very interested in this other kind of vaccines called the T-cell vaccines. So the T-cell vaccines are very, very important for long-term immunity. So what happens is, once a virus comes in and the virus gets engulfed into the into the cell, ultimately it does all its tricks, and then we have the proteasome. It takes the viral proteins and it's like a wood chipper. It takes this and then chips, like makes very, very small pieces of this protein and makes peptides. So there are thousands of peptides, as you can understand, that a virus capsid protein, like or all the virus proteins, you take them and you run them through this wood chipper, and it comes out in the forms of very, very small peptides between 8 to 14 residues. So these are peptide residues, some of which can actually be the epitopes of the virus. So the epitopes are those that actually generates immune response, right? So you have this virus. The virus, this is like a cartoon representation of the proteins that make a virus. These proteins get chipped into small peptides. This peptides traverses its way up to the surface of the cell and finds this a particular kind of protein called major histocompatibility protein, MHCs. So this peptides goes and sits on top of this MHCs, and then from there triggers what you call or invites binding to another protein called the TCR complex. The CD plus TCR, T-cell receptor proteins. And once this TCR complex gets made, that's from where the immune response begins. So these viral peptides, some of them travels, not all of them, there are thousands of them, only hundreds of the viral peptides that are actually made here can actually travel and then make these beautiful TCR complexes, which finally causes immune response. So at the core of it is a peptide protein complex. So this blue part is this protein, MHC, and the red dot here is a peptide. So this peptide protein complexes are very, very interesting. So what we have now started working on are the peptides that are coming from SARS-CoV-2. So the kind of peptides that proteasome creates after SARS-CoV-2 chips them, breaks them into small pieces. So okay, in order to do that, the first thing that we generated is what you call a tool. It's called Ensemble MHC. Again, this paper is on bioRxiv. It's under revision with Cell, one of the Cell journals. So the SARS-CoV-2 proteome is composed of structural protein, unstructured proteins, and a number of membrane proteins, envelope protein, ORF proteins. You take all of them, and there are individual websites or binding tools. So not only just binding, but binding as well as presentation. So there's a measure of presentation and there's a measure of binding. So there are like thousands of peptides generated here, 8 to 14 more peptides. We do an ensemble or a consensus modeling of this peptides binding to the MHC complexes. There are 52 MHC alleles that are available, of which there are around 64 MHC complexes, this protein, this blue protein, that are currently available in the protein data bank. So we take 64 protein structures, and to each one of them bind thousands of these peptides. And then we do this ensemble measurements or false determination rates. And then we get an ensemble binding and presentation measurement. So these are and then we find their F scores. So it turns out that the true positives are the most when we have an ensemble treatment of binding and presentation across each one of these binding tools. So our tool is nothing but an ensemble or a collection of all the available binding tools, all these ones that I have written here, and the number of true positives is most in our case. Still 50%, but we can live with that. And then we use this to identify, to basically you take, as I was telling you, there are all this HLA or MH, these are all the different MHC proteins. To that, we bind all the different small peptides coming from the surface of the virus. This peptide can be coming from the envelope protein that's in blue. So we see a lot of these peptides are coming from this ORF. So a lot of the peptides that binds to this MHC1 complex comes actually from this ORF regions. Some of them also come from the spike protein, obviously. And then once this binding is done, based on the binding affinity, we can say that, whether this protein is immunogenic or non-immunogenic. And that very recently with a company in Washington, we managed to get patient data, and we found that indeed the peptides that we were telling are immunogenic are in fact the same peptides that are triggering responses in patients. So patients that has more peptides are more immunogenic. The patients that actually have more number of these peptides coming from the structured protein or from the or from the entire virus, going up to 2,000 peptides. So this virus can generate, as I said, 15,000 odd peptides. You take the total viral proteome and break it into, divide that by 14, you'll get around 15,000. Of them, patients who have like around 600 to 2,000 peptides are those patients who are really having very strong immune responses. So the one thing that we found was that this MHC1 proteins are a very good descriptor of your immune response for SARS-CoV-2 across patients. Then we said, can we take it across countries? So we actually, this is the work that is there in the paper that I showed you. We then, there is, and in fact, India is also there in this sort of analysis. So there is available data on the website where every country has done its MHC profiling. So we know that how much population in a given country contains which particular kind of MHC. There are 52 alleles here. I can tell you that if you choose a country, and if testing has been done well there, then how much of the population contains this kind of allele versus this kind of allele versus this kind of allele. So this MHC versus this MHC versus this MHC. So we know the general population, population distribution of MHC, and based on that we can come up with a score, a mortality score, and we can correlate population mortality, deaths per million, with binding measurements coming out from ensemble MHC. And this is looking very, very intriguing actually, because in fact we end up predicting countries. So the lower the score, that means the more is the mortality risk. And we end up predicting countries. And so this is day 1, day 6, these are like days after the minimum number, after first 50. So day 0 after the first 50 deaths. So day 1 is day 1 after the first 50 deaths. It's day 6 after the first 50 deaths. And we start seeing very, very strong correlation between this binding, the bindability of MHC in a SARS-CoV-2 epitope binding inside this ensemble MHC, a molecular descriptor and a population descriptor. So for the first time, we managed to actually correlate a molecular descriptor of pandemic with a population descriptor of pandemic, and ended up predicting a lot of countries like Italy, for example, will actually see a huge mortality rate. And in fact, this was a prediction in April. The reason we started doing this in April was because at that point of time, government regulations were not in function. So it was primarily the virus versus the immune response. And that's why we wanted to get data from early April. Later on, of course, the fiduciary measures came in. There are, of course, 12 different parameters we looked into, and we found that the mortality is very, very strongly correlated with the kind of MHC1 complex present in the particular population. So therefore, by knowing what is the MHC1 setup of a particular community, of a particular, let's say, of an Indian community, of a Mexican community, of an Italian community, we'll be able to say how much risk is it, and this we can actually now take it to subsequent, basically pandemics in days to come. We can do a very similar kind of an analysis to correlate population dynamics with molecular dynamics, and we found a very nice hook, a very nice molecular hook in the form of this MHC1 complexes. So that's why I just wanted to say that a similar kind of binding studies can now help in correlating population risk with molecular properties. And so, yeah, I think, and this is a movie that kind of got made. So early on, India was there till day 15, then India vanishes. India, in fact, our mortality rate is not as high. And when we predicted back in April, we had already predicted that India will not have so much mortality. India might have infection, but not as much mortality. Countries down here will actually see a lot of people dying. In India's mortality rate today is around 2 to 3%, unlike countries lower end was around 11 to 12%. So that kind of tells that this sort of analysis works. And now we also have experimental data from patients. Good. So I think with that, I would like to conclude my talk. I'd like to say thank you to all my collaborators. This sort of work actually happens because you get to collaborate with some of the best people around the world. We have collaborators in the US, in Europe. We have students in India with whom we work, students and postdocs in India. And we'd like to kind of, continue work. This is in fact after 12 years, after my IIT Bombay days, this is my first talk in India. So I'm very, very happy that I could do this. With that, I would say thank you, and I'd open the floor for any question and invite any questions. Thanks for the opportunity again. Thanks.
Thank you, sir, for your informative presentation on resolving uncertainties of structure-based drug discovery with molecular dynamics, SARS-Cov-2 and beyond. Sir, we have two questions. The first one is, which machine learning method has been used in mining dynamics?
Yes, of course. So machine learning methods gradually are getting towards mining dynamics. There are two ways. So there is mining dynamics and there is predicting dynamics. So definitely for analyzing dynamics, machine learning has been used very, very successfully since 2008, in fact. That once you have dynamics and you want to figure out because normally molecular dynamics is very, very noisy. So you can use machine learning to figure out what are the low dimensional modes, and machine learning has been used very, very successfully in doing it. Off late, people have started working towards predicting molecular dynamics using machine learning. There also, people have showed that in the reduced space, like by using dihedrals, people can predict the dynamics of dihedrals, but they need to do something more to reconstruct the dynamics of an entire protein. But they have done it. Machine learning plus some traditional molecular dynamics tricks have been shown successful for very, very small proteins, up to 100 and 115 residues, to actually not just mine dynamics, but predict dynamics. And then, of course, as I was showing you, the third step is to integrate experiments in the prediction as well as determination of dynamics. And that's also something that's very, very recent, as recent as 2020, I would say. So yeah, so that was my response to the first question.
Okay, sir.
Yeah.
Here's one question. One more question. What type of molecular dynamics descriptors one can consider for machine learning calculations?
Yes, so that's a great question. And there is a library of descriptors. You cannot just find one descriptor. There is a library of descriptors depending upon the problem. And therefore, and so people had asked this question in different ways and they have given up on this question. And so therefore, what they have done nowadays is they have said, okay, I will not figure out what these descriptors are. I will let my computations, I will do a machine learning algorithm to actually determine what these descriptors can be for me. So there are methods like pseudospectral methods that can be used. But I would say in a general sense, people have used, there's something called VAMPNet. So descriptors can be some large scale collective variable, but which often depends on the intuition. It can be protein-protein contacts. If you are very, very aware of what your system is, you can find protein-protein contacts. You can find distances. Those are very, very intuitive. But once you have those, even those, I would say distances and dihedrals are often two variables that are very, very interesting. People have found a lot of success with that. But after that, people have seen that even those are not working as well. So then they said, okay, fine, we will let machine learning do the work for us. And there are things like neural networks where you can basically construct a reduced space by using, as I said, tools like VAMP nets, which will give you your neural network, your low dimensional representation.
Thank you, sir. for your elaborative answers. Now, on behalf of Director CSI NIST and DDH Training Program team, I would like to thank you for accepting our invitation and give presentation on this platform. Now, with this one, we are concluding this session, sir. Okay. And I request all the participants to join on Vertical 3 at 6 p.m. Thank you, sir.
Thank you.